Thanks for coming. First of all, I want to thank the Hamilton Committee for giving me the opportunity to speak today. I'm going to be talking a little bit about my work in the Research lab that I was working on with Sally Otto and Rose Ann. Okay, so like many people in this room, I'm interested in organismal diversity, and in particular, why that diversity is sort of discrete and not continuous. So another way to think about that question is, I'm interested in why there are species. And what that means is that I'm interested in the process of speciation, which can be thought of as the evolution of reproductive barriers, which are these traits or mechanisms that reduce the ability for different species to reproduce and or to produce successful offspring. And there are a lot of really interesting questions that can be asked about reproductive barriers, like how and why they evolve. But today I'm going to be focusing on the question, which reproductive barriers actually cause speciation? And at first this seems like a really straightforward question, and in a lot of ways it is. But there's a bit of a problem in that by the time that populations have diverged enough for us to consider them species, a lot of times many reproductive barriers have evolved, and at that point it becomes really difficult to distinguish the barriers that actually cause speciation and those that accumulated once speciation was largely complete. However, there's a nice trick to get around this, which is to try to measure reproductive barriers between really young or incipient species. And we have a nice example of that within the species Helianthus pigularis. So this is a widespread sunflower species that typically occupies sandy soils throughout the central US, but there are populations of this sunflower that grow at Great Sand Dunes National Park in Colorado, where there are the tallest sand dunes in North America that reach up to 200 meters tall. These populations look like this, and as you can imagine, it's a pretty harsh environment for plants to survive in. Okay, so the important thing about this dune and non-dune ecotype within Helianthus pigularis is that from work that Rose Andrew did, we know that using RADSeq and microsatellite data, as well as the glacial history of the area, we can be pretty sure that these ecotypes started to diverge less than 10,000 years ago, potentially quite a bit less. In addition, we know that there's ongoing gene flow between them, but that there are a few large regions of genomic divergence that separate them. And when you take all of these traits together, it seems like these are a pretty good candidate for incipient species, and a really good place to look for early arising reproductive barriers. So when you are doing this type of study where you're looking for reproductive barriers, it's really important to measure as many as you possibly can. And during my PhD, I managed to get to 7. These were selection against immigrants, flowering time differences, differences in pollinator communities, pollen competition, hybrid germination, hybrid fertility, and selection against hybrids. And also, what's really great about this list of barriers is that it includes both those that act before zygote formation and those that act after, which is interesting because several studies and meta-analyses have suggested that pre-zygotic barriers are more important during early speciation than post-zygotic barriers, so we have the opportunity to see if that's true here as well. And in addition, they include both extrinsic reproductive barriers, which are those that are environmentally dependent, and intrinsic barriers, which are environmentally independent. And again, in a case where it looks like local adaptation is probably going to be really important, we might expect that extrinsic barriers would be stronger than intrinsic, or that maybe we won't find any intrinsic barriers at all. Okay, so getting straight into measuring reproductive barriers. The first thing that I did was a simple reciprocal transplant. This allowed me to measure both selection against immigrants and selection against hybrids. And I also just wanted to note here that the major phenotypic difference between dune and non-dune sunflowers is that the dune plants have seeds that are about 3 times larger than the non-dune plants. So at the same time, we were interested in how this trait might be involved in these reproductive barriers. Okay, so we planted dune, hybrid, and non-dune seeds out into the non-dune environment that looks like this, and into the dune environment, which is quite impressive. And what we find when we look at our first component of fitness is that seed weight is strongly correlated with seedling emergence in the field. So in this graph, you can see seed weight along the X and emergence on the Y, and there's a pretty obvious correlation there. This This relationship is the same in both environments, so both in the dune and non-dune habitat, and it means essentially that dune plants end up emerging better than non-dune plants, and hybrids sort of span the range in the middle. Skipping over the survival data for a minute, when we look at the number of seeds that the surviving plants make in all the habitats, what we find is that non-dune plants make many, many more seeds in the non-dune environment than any of the other trait and habitat combinations. Okay, so when we take the fact that large dune seeds emerge better in both environments, and that non-dune plants make many more small seeds in the non-dune habitat, and also throw in the other measures of fitness that we took, we find really clear evidence of local adaptation, where the dune plants do better in the dune environment, non-dune plants do better in the non-dune environment, and hybrids sort of don't do well really anywhere. And this pattern of local adaptation leads to strong selection against immigrants and strong selection against hybrids. So in this graph here, eventually I'll be listing out all the reproductive barriers along the X, and the strength of reproductive isolation is on the Y, where reproductive barriers of 1 mean that there's pretty much complete reproductive isolation, and 0 is essentially random mating. The next reproductive barrier that we measured was flowering time. I did this both by looking at the date of first flower during the reciprocal transplant and by putting time-lapse cameras out into the populations to look at flowering time more generally. And basically what we found is that any way we looked at the data, there were no differences in flowering time, and that leads to a very weak reproductive barrier. We also surveyed pollinator communities in both habitats by netting insects that were actually visiting sunflowers and also using pan and malaise traps. Then we separated all the specimens out into morphospecies and sent the bees away to be identified to species. And what we found here is that even after taking distance into effect, there's very different pollinator communities in the dune habitat compared to the non-dune habitat. And then when we look at the visitation rates to sunflowers of the pollinators that are shared between the habitats and those that are not shared, we find a very strong reproductive barrier. However, it's important to note that this is probably actually an underestimate, because for the pollinator species that we find in both habitats, we don't actually, even though we know that they're in both habitats, we don't actually have data that shows us that individuals actually move between the two habitat types, which may or may not happen. We also looked at this barrier called pollen competition, which arises when two types of pollen arise on a stigmatic surface at once, but then there's a non-random, it's non-random which type of pollen tends to fertilize ovules. And it's important to note here that when you make crosses between the different ecotypes by themselves, they set seed just fine. So the way that we measure this barrier is by mixing dune and non-dune pollen and taking that mixture and pollinating it, using it to pollinate multiple moms. And when we do that, we get data that look like this. So here we have the proportion of offspring sired by dune plants on the Y, and all the different mothers we phenotyped along the X, and they're color-coded by their ecotype. And the take-home message here is that we find strong sortative mating, where dune moms tend to set seed sired by dune dads, and non-dune moms tend to set seed sired by non-dune dads. This assortative mating leads to another reproductive barrier. And finally, we looked at hybrid inviability and sterility by looking at seed germination in the lab, as well as looking at pollen viability in the lab. And what we found here is that both hybrid seeds and hybrid pollen is as viable as either of the parental types. So this means that there's essentially no reproductive barrier here. Okay, so looking at all of these barriers together, we can see that there are quite a few strong reproductive barriers. And then when we break this down a little bit again, we find several strong prezygotic barriers as expected, but we do find evidence for one postzygotic barrier, specifically extrinsic selection against hybrids. We also find several strong extrinsic barriers, as expected, that seem to be directly related to local adaptation and or moving into a new habitat type. However, somewhat surprisingly, we found this post-pollination assortative mating via pollen competition. And the future directions for this work is to really dig into this particular reproductive barrier and try to figure out which evolutionary forces are responsible for it. Okay, so now finally, when we go back to our original question of which reproductive barriers cause speciation, I think that what these data are showing us is that multiple and diverse reproductive barriers can arise really quickly, even during the earliest stages of speciation. With that, I'd like to thank you all for listening, and I would be happy to take questions.